Hello, everybody. Welcome to our live. All right, welcome to our live, everybody. We are going to get our co-host going quickly. Thanks for coming. Hi, everybody. 48 hours until the exam.
Hey, everybody.
How are we doing? Happy Saturday.
Happy Saturday.
48-hour countdown.
48-hour countdown. There's a sound question. Wonder who she can't hear.
Oh, no. Stop it.
Don't you dare. Not happening today. I can hear you. We're good on my end. I hear you.
Can you hear me?
I hear you.
All right, let me
So
try.
I think we're okay.
Can you still hear me?
I can still
Awesome.
hear you.
Okay.
And you hear me, apparently.
Yeah.
So I think we're good.
For those of you who don't understand why we're asking these questions, we crashed our Instagram Live on Thursday. Or I crashed it, and then we had to restart.
Miss Coffin's here. That's great.
Hi, everybody. Yeah, so where are you from? I am Melanie Kingett. I'm from the APsolute recAP, and I'm currently in Pennsylvania.
And I'm Tiffany Jones, AP Bio Penguins, or just a normal teacher. Oh, I'm in Georgia.
Awesome. Okay, so if you are new to Instagram Live or it's your first time joining ours, couple things. You see that there's the comment box, obviously, but there's also that little question mark. Please feel free to submit your questions there. That way they don't get lost in the comments and we can still get to those. We're going to talk about a lot of strategies today, review as much content for you as possible. However, it might be more of a skimming, right? Because we want to get to a lot of topics and not necessarily dive super, super deep because time is of the essence. All right, so let's kick it off with our first question.
We've already got questions? Yes.
Yes, hold on. All right, it looks like one of my question boxes is not popping up. So we're going to wait. So if you guys have questions, please pop them into the box. We did have a question that came in previously today that was how to distinguish between describe and explain. I know you talk to your students a lot about those task verbs.
So for me, describe is where you're just giving details. So depending on whether they're giving you a diagram and you're using that diagram to describe, you can use the diagram, explain things that you see in I use the other task verb in that answer. So you'll use information from the diagram to be able to say why the answer is what it is. Versus explain is the explain how and the why at the same time.
Yeah.
So they are very similar. And kind of as long as you're detail oriented, you'll be fine on either one of those.
Yeah, details are where it's at. Definitely do not. I'm gonna put this one out because I think it's really funny.
Sorry, that might be an honor code violation.
Might be.
But you can have some penguin power.
Yes, awesome. And guys, it's not about knowing all the answers. I had mentioned this one of the other times we had a live, but like people who go on Jeopardy, they don't know everything. They can just really pick up on context clues. So use your diagrams, use the prompt and the FRQ, and extract information out from that can help you answer the prompt.
Especially this year with it being an online exam and with them allowing you to have open notes and technically open internet because
I know, right?
You will be able to get everything from that prompt. There's probably very minimal questions that are straight content. It's going to be application of content. And so you'll have to rely on your biology knowledge to understand why we're seeing what we see in the data or why we see what we see in the prompt and the explanation of the scenario.
Yeah, especially FRQ2, right? So FRQ2 is about a disruption to something that would normally happen. So you have to be able to reflect back on why something has changed and what is specifically happening with probably the molecular chemistry to have that change have the effect. All right, so a good starter question. What can pass through a permeable membrane? All right, so
Taking it or not taking it?
I'll start it and you kick in. All right, so let's remind ourselves what membranes are made of. So we have our membranes made of phospholipids, which have their hydrophilic and hydrophobic regions with embedded proteins and cholesterols and all of that. But that means it's semi-permeable, right? Some things can pass and some things cannot. So large and charged, those are your big no-nos, okay? Small, and specifically, typically like hydrophobic, they can pass through. So like your gases can pass through, thank goodness. Water is really small. Now I know water is polar, but water is really small. So in individual molecular quantities, it can pass through, specifically through osmosis. If you got to move a lot of water, you're going to go through a protein and aquaporin. What about active transport, Tiff?
Active transport is more looking at going against the gradient. And so these are going to require some type of transport protein. You're going to have to either use a carrier protein, and you probably won't use a channel protein because of the way that they're set up. And you're going to have to use ATP. So ATP is where you're going to see a exergonic reaction. Somebody asked earlier about exergonics. I figured I'd slip that in there. And so when ATP breaks down into ADP and it phosphorylates that protein, it makes it primed to do work. And so now you can see that the protein will change shape, which will carry the material across the membrane. So it'll allow us to pump something against the gradient.
Yeah, and so we're going to use the proteins during facilitated diffusion as well. But remember that it's directional movement. So facilitated diffusion still uses the protein, but we're going to move from high to low concentration in that context. I'm just moving through the scroll right now in comments. So I see a couple questions that popped up in there. Just remember, guys, if you guys can put them in the actual question box, that'll help us not to lose them through all of your chat. Oh, good. Turkey can join us again, our follower from Turkey.
I figured that would be helpful with her since it wouldn't be as late.
9 p.m. and not 3 in the morning. We appreciate the dedication, though. It's really awesome. I like this question because it's about how specific, right? Do we have to be, or is a general understanding sufficient?
I will tell you. Sorry.
No, it's a hard question to answer.
So I believe it was about 4, maybe 5 years ago. There was a question on cell respiration, and they actually gave you the 3 major steps of cell respiration, and then they asked you questions about it. And so you were doing more of an application of it. Why do we have NADH? How is glycolysis allowing us to make ATP? And so we all know that's substrate level phosphorylation, right? And then also the NADH was going to electron transport chain, providing the electrons so that you could go through oxidative phosphorylation. So like it's about more of an application process. So I would have a general idea of the steps and then be able to apply them to why things are happening.
Yeah, I agree with that completely. It's not about knowing what the steps are in so many ways, or the specifics. It's about why and how does that molecule have a role? Like, what is the role of each molecule in the process? So it's a really, really great thing. And the same can be said about photosynthesis, right? I mean, we look at those diagrams of the Calvin cycle and the Krebs cycle, and our brain starts to get a little loopy. But you're not required to know specific enzymes. You're not required to know those intermediate molecules. But an understanding of why and how the creation of them and what going through the cycle does for the process is important.
So I always tell my students it's important to know what goes in, what goes out, and why is it important.
Yeah.
So like I like my students to know what is the starting material for glycolysis, what's the starting material for Krebs cycle, what is the products of those kind of. It's important to know that oxygen is that final electron acceptor and that you're making water. Like that's an important component of it.
Super.
But you didn't have to know specifically which cytochromes are in the electron transport chain and how the process takes place.
And then and then recognizing that role of water in photosynthesis, right? So you have that bookend of water being split and donating electrons to the chain in photosynthesis, whereas when you have your cellular respiration, you get water at the very end when electrons exit the chain. Okay, I like this question too, because again, it's not about content. You guys are at skill-based components here. So how do you suggest we approach data and analyze it accurately? Whew. So we mentioned this yesterday. No, what's today? When we're home, everything blends together.
2 days ago.
Thursday, we talked about, I know it didn't get saved, but we talked about how to approach an FRQ. And I think you and I both tell our students very similar things. Jump to the questions first. Some of them don't even require you to read the prompt to answer. But if you know what the questions are, and this is not a strategy unique to biology or unique to this exam, this is a general test taking strategy. If you had a literature prompt, this is also useful. Look at your questions, then go back and look at your prompt. But what would you say about those data tables?
They're going to try to trip you up a little bit. So a lot of times those error bars, they're specifically made so they will overlap. So it appears like your data is increasing or decreasing, but in actuality, with the error bars overlapping, the data is not statistically significant. The big example that comes to my mind is the caffeine B question from, I believe, 2017, in which short-term memory was not statistically significant because those error bars overlapped, but long-term was. And like both of those did look like they were increasing because of the means, but it had to do with those error bars. Also, when we looked at the FRQ we did on Thursday, they flipped the axes. So it's important that you understand what the difference is between an independent and a dependent variable. And you're not just going to say, oh, well, the one on the x-axis is my independent, the one on my y-axis is dependent. You have to actually understand the question and kind of look at the actual experiment that they're doing.
Yeah, don't jump to
I answered their
conclusions
question.
and don't make assumptions about your data. I think that could be a real big trip fall. So when you look at those graphs and you want to see things as being statistically significant, they're going to want you to really use that word because that's going to show your understanding of error bars and your understanding of why the data may or may not reject the null hypothesis.
One more thing about those error bars, because I don't know how many of you guys are return people, but I'm an AP reader, so I actually have graded FRQs on the exams. And so one of the things about those error bars is that you're specific about which error bars. So there may be 4 error bars in your graph, and 2 of them are what you're using to state statistically significant. Be sure to explain, I use, 10 minutes control, 10 minutes caffeine, or however specific examples you're using, which error bars are you analyzing? Because just saying error bars overlap is not detailed enough.
Absolutely not. Especially when they're going to probably have 2 graphs on there as well. And I'm going to pop this question up here specifically because
I had one more thing to say.
Oh, sure. Yeah. I'll put this one back up in a sec.
You're good. So a couple years ago, there also was a mouse question, and it had this weird anagram about their awake and sleep cycles. A lot of times with these graphs, if you just tell them what you see in the graph, sometimes that is a point. And so being like and it may be like an obvious thing. I saw that they were asleep for 12 hours and awake for 12 hours. That could be as simple as your answer. So don't overthink your graphs whenever you look at them. So you can put your question back up now. I'm sorry.
Yeah, you have like, you're like an FRQ encyclopedia over here.
Sorry.
All right. So what is the difference between a null alternative hypothesis? This tends to get overcomplicated. It's just fancy wording for something you already understand how to do. So null means not, generally, right? So in a null hypothesis, it's when you don't expect to get results. So if you're testing the significance between certain variable groups and there is no statistical significance, then you don't get a difference between them and you can't reject your null hypothesis. I said that really backwards, but it made sense in my head. Can you translate?
I can translate or try to translate for you. So what we're saying with the null hypothesis is that the treatment had no effect on the results. And so that's where that statistically significant part comes in. And so we would say that we reject the null hypothesis or we fail to reject the null hypothesis. Versus your alternative hypothesis is just saying, I believe, like, what is going to happen in the experiment. If you add caffeine, the memory will increase in the B or whatever it is.
And so when you're going to look at the data to reject or fail to reject is going to come down to your chi-squared value, right? So if your chi-square, if it's larger than your critical value on your table, then you're going to reject your null hypothesis because there is statistical difference between your groups. It's not just due to chance versus the other way, which just means you fail to reject. Okay. A lot of tongue twisters in there.
It is.
All right.
And remember to have your formula sheet, because that chi-square chart, the critical value chart, is on the formula sheet. So you can either have it pulled up on your computer as like a split screen, or you can actually have it printed out and next to you, whichever is best for you taking this exam.
So that question was, how many parts are in each question? It's less about the parts, more about the actionable verbs that you have to do: describe, explain, predict. Those are where your points come from. And we want to make sure that you address each one of those. So the sample that we went over on Thursday was A through G, I believe. And that's just because A through J. So they broke up all of the verbs into independent questions, whereas in some of the other samples, they were smushed together. So even though it was 4 parts, part A had 3 things you had to do. So we're looking at probably 10 points for FRQ1, probably 6 points for FRQ2, but that hasn't been specifically stated, has it?
No. And if you do the math, 10 divided by 16 is 62.5%, which is close to that 65%. But they can also just use multipliers to get to the 65% and the 35%. There is a question that's asking about, is accept the same as fail to reject? So some years we've been able to accept hypothesis. I mean, fail to reject was the same as accept. In some years, it's not. So it's really hard to say whether that would be an appropriate thing. It's all, I'm going to be completely honest with you, it goes down to the question leader. So when we get to the read, the question leader sits down and they look through thousands of responses and they kind of see what the students are writing and how things are going. And that's when they develop the actual rubrics for these for responses.
I like this question because it's about preference. Do you suggest to handwrite or copy and paste? So let's just review the 3 choices that you have. So when you're submitting your responses or answering your questions, you cannot type them directly into the program. So you have to have either a separate document that you're writing, or you're writing them by hand. Your 3 submission choices are to copy and paste. That would be a digital. You can upload a text file, a doc format, a PDF, or you can take a picture of your handwritten responses and upload those as well. So those are your 3 choices. I think copy and pasting is the easiest. However, you should practice multiple ways so that you can see what's most comfortable for you. Just be mindful that when you copy and paste, any like automatic formatting might not transfer over. So in like Microsoft Word, if you were to use the like numbering function to mark your responses, you just want to make sure that that transferred properly and you wanted to express it. I wouldn't use any automatic numberings if I were you, but you got to practice. So use the demo that has been put out by the College Board. Make sure your tech works. And
update your browser.
Update your browser. And if you're an iPhone user, make sure you change your file format. You don't want to use the default picture format, which is HEIC. In your settings and go to camera, you change it to most compatible. That way, when you take a picture of your answers the day of the test, those will upload for you. But you do have to upload from exactly the same device that you access the test question. So if you're on a laptop looking at the question and take a picture with your phone of your handwritten work, you do have to transfer it, which can take extra time.
Also, there is if you're one of these students who does this, that you handwrite your response and then you save it as a PDF, like you do like a scan function with your phone. That's actually considered option 2 because you're uploading a document.
I love this question too. Do we lose points for questions we don't answer?
Nope, you're adding points, and this is a points game. So if you don't know the answer to A, skip A, go to B, do C. Oh, you don't know D? Skip D, go to E. Do what you know first, and then come back to the parts that you don't know. And the good thing this year is you only have 1 question at a time. So you know you have 25 minutes to sit there and make sure you can come up with whatever you can for any part of part 1. It's not like when you traditionally have all 6 questions and you have to decide how to, divvy up your time. You only have this 1 question for 25 minutes. So put everything you got at it and nothing blank.
Yeah, nothing blank. So you are not going to be making a graph yourself. You yourself you are not graphing, but there will be graphs, and most likely in FRQ1. What kind of graphs? Oh, it's so varied.
It can be a line graph, it could be a bar graph, it could be a scatter plot, it could be a box and whisker. I've seen the anagrams. They can sometimes pull graphs that you may have never seen before. But if they give you something that's not directly in the standards, they have to tell you what it is and kind of give you background information about it. So like the anagram that they had in 2012 exam, I believe, is it kind of told them that the darker spots was when they were awake and lighter spots was able to get a 5 even if you don't answer everything. Oh no, what's happening to her? Are you there? Oh no, did it happen again? Can you guys hear her?